Hello, everyone, and welcome back to today's practice session. And the topic of today's practice session is molecular basis of inheritance. So, first, let me check if I'm audible or not. The most obvious question, right? The most obvious question, the most asked question these days. So, yes, I am audible. So, guys, I welcome you all to your one and only favorite channel, Let's Crack NEET UG in English, where we are continuously providing you live lectures of all the three subjects, right? Where we are continuously providing you live lectures of all the three subjects. And I'm also providing you the live practice session so that we can enhance our problem solving ability in biology also, right? Because we do solve questions, we do solve a lot of questions when we study physics or chemistry, but when it comes to biology, we read the theory part and then we feel like that's it, we'll be able to solve the questions. Yes, theory part is needed for the conceptual clarity, but at least we need to know what sort of questions are being asked from any chapters, okay, or from the entire biology. And for that only I'm trying to bring these various practice sessions, okay. This will definitely help you in enhancing your problem solving skills and in applying the concepts which you have learned in the theory classes, okay guys. So come on, let's start today's session without wasting any time. So, first of all, as a ritual, let me first introduce myself and tell you something really important about Unacademy, and then we'll be starting with our practice session. Okay, guys. So, this is the Telegram channel link. You can stay connected to all the educators of this particular channel through this, Telegram channel. Okay, you will be getting all the updates of the upcoming lectures through this channel. Now, talking about myself, so my name is Aditi Gupta. I'm your Aditi ma'am, and I have been mentoring students for the past 6 years for their NEET UG preparation. And I myself have secured an All India rank of 61 in the prestigious GATE examination. Okay, guys. So as you are well aware of the fact that yeah, there are topmost team of educators on board on this awesome platform called Unacademy. But to avail maximum benefit, you need to go for an Academy subscription. You can download an Academy learning app from Play Store or iOS, and this learning app contains some absolutely free of cost features also. For that you don't have to pay any fee, right? And those free of cost features include free live classes and the quiz features. Okay. Talking about my timings on an Academy learning app, I'm live daily at 11 a.m. You can find my profile link in the description box. Then talking about the Unacademy subscription, so guys, you will be getting live classes from the topmost team of educators. You can select your batch as there are multiple batches running on the platform, and you can track your preparation with the help by participating in various test series which are undergoing, right? Now, which are ongoing, right? Now coming towards the platform part, so there are two platform on Unacademy subscription, Unacademy Plus and Unacademy Iconic, right? So talking about the Unacademy Plus subscription, so guys, on Unacademy subscription there are 50 plus educators available on board. Okay, now you will be getting daily live classes of all the three subjects, physics, chemistry and biology, on Unacademy subscription platform. And you will be getting a chance to interact with your educator one on one. You will be getting doubt discussion sessions. Okay, then the complete NEET UG syllabus will be covered in a detailed manner, in a thorough manner, along with the PDF notes of each and every chapter. Okay, then your performance will be evaluated on the basis of live tests and quizzes so that you can check whether you are able to solve the questions or not. Then you will be getting unlimited access, that means the access to the video lectures, to the PDF notes, to the personalized test papers of all the educators available on Unacademy subscription platform. Okay, so these are the benefits of Unacademy Plus platform. Now coming towards the subscription fee. So an Academy Plus comes with various options. These shorter duration are for crash courses generally. But I would suggest if you're seriously preparing for NEET UG examination, then 1 year or 2 year of subscription time is required, or preparation time is required, and to prepare effectively for NEET UG examination, right? Those students who are in the mid of the session preparing for NEET 2021 can also opt for the 6 month of subscription plan. Otherwise, 10 pass out students should go for 24 months, and 12 studying or 12 pass out students should opt for 1 year of subscription plan. Okay, guys. Now, if you use my referral code, which is AG001, then you will be getting an instant 10% discount on your course fee. That means you will have to pay now rupees 25,200 for 6 month of subscription plan, 34,650 for 12 months, and 50,400 for 2 year. And do compare the monthly cost which you are paying. For example, in 2 year subscription plan you are paying only rupees 2100 per month, but see, if you individually buy a single month subscription, you have to pay rupees 7,000. So the subscription fee has been reduced many falls in in a longer duration subscription plan. Now, coming towards the iconic platform, so see, this iconic platform is the brand new platform introduced from Unacademy, right? And it includes some out of the box features. It already has all the benefits of the Plus platform. Okay. Apart from that, it does have personal coach. Each and every student on Unacademy Iconic will be having their own personal coach who will be there to listen to you, to listen to your challenges, to listen to your problems. Okay, will be there to guide you, to keep you motivated during your NEET UG preparation journey, right? Then you will be getting a study planner so that you can utilize your time to the best of your efficiency. The best feature is the test analysis. That means you'll be getting in depth test analysis. You'll be learning from your mistakes, and these mistakes will help you in improving your problem solving skills. Okay, you will be knowing what sort of mistakes you generally do while you appear in a test, right? You can go back and rectify them so that you never repeat those mistakes again. And all the benefits of Plus are included, and also from time to time you will be getting expert guidance or expert motivational session, right? So coming towards the subscription fee part, guys, there are three subscription plan available: 6 month, 1 year, and 2 year. Okay, again, if you use my referral code AG001, you will be getting an instant 10% discount. That means the fee which you have to pay now is rupees 34,200 for 6 months, 58,500 for 1 year, and 90,000 for 2 year. Okay, the monthly cost is rupees 3,750 for this 2 year of plan. At this 3,750, you are getting all the benefits of the iconic platform. So you know this is the cheapest and the best available plan. Right, guys? So do not delay your subscription. Go buy your subscription and start your preparation from today. Okay, guys? And do not forget to use my referral code AG001. So come on now, let's start today's practice session, guys. So again, this practice session have approximately 40 questions from this particular chapter, molecular basis of inheritance. In short, we also refer to this chapter as molecular genetics, right? You know, this is a really important chapter from class 12 NCERT. And I have tried to include the basic question and also some good questions in this practice session. Although we will be organizing multiple sessions, okay? So of small lots, right? So today is the first day of this practice session. So this is the first question. I'm starting with a very basic one. So in a DNA strand, the nucleotides are linked together by glycosidic bonds, phosphodiester bonds, peptide bonds, or hydrogen bonds. You know the rules. You have to mention the question number and then mention the options, like it is 1, 2, or whatever is the option. Okay, so do mention the question number and try to participate in the practice session. Do not hesitate while answering, because the most important thing is to participate. Do not worry if you're not able to solve it right, because you are here to learn. Okay, so try and participate in the session. So in a DNA strand, the nucleotides, we know that the nucleotide is a structure which is made up of a pentose sugar. On the first carbon, nitrogen base is present. On the fifth carbon, the phosphate group is attached, the phosphate ion is attached. And two nucleotides, they are connected with the help of this phosphate ion which is attached to this fifth carbon, right? And it forms a phosphodiester bond with the help of the oxygen present at the third carbon. This is the first carbon, this is the second, this is the fourth carbon, this is the fifth carbon. So this bond is what? This bond is the phosphodiester bond. One ester bond is present over here, another ester bond is present here. So it is phosphodiester bond. So right option is option number 2. Right? Moving ahead. A nucleoside differ from a nucleotide. It lacks. So you have to identify the structure which is missing in a nucleoside, but it is present in a nucleotide. So what is the structure? Is it base, sugar, phosphate group, or hydroxyl group? You know that nucleic acids are polymer of nucleotide, right? Nucleic acids are polymer of nucleotide, and a nucleotide is made up of nitrogenous bases, nitrogenous bases, pentose sugar, along with the phosphate ion. So this is the nucleotide. Now, which one of the following structure is not found in a nucleoside? So a nucleoside consists of the sugar molecule along with the nitrogenous bases. Along with the nitrogenous bases, that means the phosphate group is missing. So the right answer is option number 3. Okay, right answer is option number 3. Very good, Kunal. Absolutely correct. Yes, I understand, because I have covered the biomolecule in detail. You must be knowing the detailed structure of the nucleic acids. So you will be able to solve these questions. Now moving ahead. Both deoxyribose and ribose belong to a class of sugars called, is it trioses, hexoses, pentoses, or polysaccharides? So I have already shown you the structure of the sugar. And I have already shown you the number of the carbon atoms present. So there are 5 carbon atoms present, right? 1, 2, 3, 4, and 5. One is present outside the ring. So that means, there are 5 carbon atom in this sugar. That means this belong to pentoses. This belong to pentoses, right? So the right answer for question number 3 becomes option number 3. So 3 and 3 are rhyming. So moving ahead. Question number 4. Next question is, the fact that a purine base always pairs through hydrogen bonds with a pyrimidine base in a DNA double helix leads to the anti-parallel nature, the semi-conservative nature, uniform width throughout DNA, uniform length in all DNA. Okay, so you know that in a DNA molecule, the purine, it always base pairs with a pyrimidine molecule. Okay, this is a purine. Okay, bicyclic structure. It always forms pairs with a, it always bonds with a pyrimidine molecule, right? Always. For example, if this is adenine, okay, if this is adenine, then this becomes thymine, okay, and they form double bond, right? Similarly, if there is, guanine, if there is guanine, okay, it will be forming a triple bond with cytosine. It will be forming a triple bond with the structure of the cytosine. So every time a purine is binding with a pyrimidine, so this is, maintained in the structure of DNA, so that the width of the DNA, remember I told you that the diameter of the DNA is 20 angstrom, and the diameter means the distance from phosphate to phosphate, right? So, the in order to maintain the diameter constant, the it is always that a purine always base pairs with a pyrimidine. That means uniform width throughout the DNA. So the right answer is option number 3. Okay. Now, moving ahead, yes, Kunal, this is right answer. Option number 3 is the right answer. Next question. The net electric charge on DNA and histone is, okay, both positive, both negative, negative and positive respectively, or 0. So guys, you have to identify the charge on DNA and histone. And this we have discussed multiple times. I've told you that the charge on the nucleic acid, be it DNA, be it RNA. The charge on the nucleic acid is negative, and the reason for this negative charge is the presence of the phosphate ion. And I've also told you that these histones are the basic proteins. Okay, basic proteins. Why? Because they contains basic amino acids, that is lysine and arginine, in more numbers. These lysine and arginine, these are the basic amino acids. So they are present in more amount or more numbers. So they are basic proteins, that means positively charged proteins. So the right answer to this question is negative on DNA and positive on histone. So the right answer is option number 3. Okay, very good. Next is, the promoter site and the terminator site for transcription are located at. You have to identify the promoter and the transcript the terminator site. 3 prime downstream and 5 prime upstream and respectively of the transcription unit. 5 prime upstream and 3 prime downstream and respectively of the transcription unit. Then 5 prime upstream and then 3 prime downstream end. You have to identify the promoter site and the terminator site. While answering this question, do consider this in mind that these 5 prime or 3 prime upstream or downstream is referred with respect to the coding strand. We know while during the transcription, the 3 prime to 5 prime strand, this strand behave as the template strand. Remember, this strand behave as the template strand. RNA polymerase, it binds in front of this 3 prime end of this strand, and it starts polymerizing the RNA in this direction, 5 prime to 3 prime direction. But the coding strand is this one, right? And the references which we made is with respect to the coding strand. So if this is the structural gene, that means which contains the information. So now promoter is present upstream and terminator is present downstream of the structural gene. That means 5 prime upstream and 3 prime downstream of the transcription unit. So second option is the right option. Next question. Which of the following statement is most appropriate for sickle cell anemia? Okay. It cannot be treated with iron supplements. It is a molecular disease. It confers resistance to acquiring malaria, or all of the above. So which one of the following is the most appropriate statement for sickle cell anemia? So guys, you remember this sickle cell anemia because of the mutation. Because of the mutation at the sixth position, what happened? Valine amino acid was formed, right? Now we know that the sickle cell anemia, it is a hereditary disorder. It is a hereditary disorder. It cannot be treated with iron supplements. So this is a right statement. It is a molecular disorder because it involves mutations, right? It involves mutation. Now it confers resistance to acquiring malaria, yes, because the RBC is sickle shaped. When, see what happens, the hemoglobin is, the condition of the hemoglobin is HbS. S stands for sickle cell, and whenever oxygen is not present in sufficient amount, so what happens, the sickling of RBC starts to take place. And the sickled RBC are are not a good host for this malarial parasite. So all of the above options are correct. Option number 4 is the right answer. Very good, Kunal, absolutely correct. Question number 8, which of the following is true with respect to AUG? Okay, it codes for methionine only. It is an initiation codon. It codes for methionine in both prokaryotes and eukaryotes, or all of the above. What do you think should be the right answer with respect to this AUG codon? Okay, with respect to this AUG codon, I appreciate the efforts of those students who are regularly participating in the practice session and are regularly you know present, because by participating only you'll be able to know your knowledge, you'll be able to evaluate yourself. Okay, so do not hesitate while answering the questions. It may be right, it may be wrong, but it doesn't take anything, right? If in case if you're wrong also, but then also you are learning something. So do participate in the question. Okay. So codon is the is the arrangement of nitrogenous bases, and that too in the triplet manner. Okay, for example, adenine, uracil, and guanine. They are present in the RNA. The information from DNA is transferred to the RNA molecule, right? Now, what happens in the RNA, in place of thymine, uracil is present. In the RNA, all the nitrogenous bases which are present, they are always read in triplet manner. And these triplet information are the codon. Okay. So now what happens? This AUG is a codon which represents methionine. Yes. And it codes for methionine only. Yes, definitely. In case of eukaryotes, in case of prokaryotes, it represents formilated methionine. But again, it is methionine only, right? So this statement is correct. It is an initiation codon. Yes, always the process of translation starts with methionine, that means AUG codon. Now, it codes for methionine in both prokaryotes and eukaryotes. Yes, it is also correct. Do not mistake it that in prokaryotes, definitely it codes for formilated methionine, but it is a version of methionine only. Okay, so this statement is also correct. Because we know that the genetic code is universal in nature. It is not like we'll be having an entire different set of genetic code and different organism will be having a different genetic code. No, it is universal in nature. We have read, this is one of the properties of the genetic code. So all of the above option is correct. Okay, so very good, Karthik. Yes, absolutely correct. Moving ahead, the first genetic material could be protein, carbohydrates, DNA, or RNA. So, while taking classes for molecular genetics, I've taken this class, a month ago, and I told you this story of the evolution of the first genetic material. So, what is the, what was the first genetic material? Was it protein? Was it DNA? Was it RNA? First of all those students who have studied molecular genetics, they very well know that protein are not the genetic material, and we have seen how beautifully different experiments were conducted to disprove this concept that protein is a genetic material, right? We know that yes, DNA is the genetic material, but the question is asking the first genetic material. So guys, remember RNA world. So RNA world was the dominant world, right, at that time. So the right answer is RNA. The first genetic material was RNA, and from this, this RNA, it has dual property. It has the property of acting as a genetic material also, and it had the property of enzyme also. It had enzymatic activity also. So what happened with this RNA molecule was able to replicate itself, and also because of its enzymatic activity, with the help of amino acid, it synthesized the formation of proteins, right? Then what happened in the transition world? Now what happened in the transition world? This RNA got converted into DNA molecule in the presence of the protein. And what is happening in the present world? The DNA is replicating itself with the help of the protein. DNA is converting into RNA with the help of the protein, right? And RNA is RNA is transferring its information into protein again with the help of protein, right? So the first genetic material is RNA. The first genetic material is RNA. Yes, DNA is a stable genetic material and DNA is the genetic material, but the original one, the first one which was formed, was the RNA. Question number 10. With regard to mature mRNA in eukaryotes, exons and introns do not appear in the mature RNA. Exons appear but introns do not appear in the mature RNA. Introns appear but exons do not appear in the mature RNA. Both exons and introns appear in the mature RNA. First of all, you need to know what are exons and what are introns. Okay, so again, after knowing this information, the question becomes really easy. Exon is the functional part. Exon is the functional part. Okay, and intron are the non-functional part. Right. So what happens in case of mRNA in eukaryotes? You know what happened? The pre-mRNA, that is the hnRNA, it does contain both exon and intron, but the processed RNA, that is the mRNA, it do not contain the intron. It do not contain the intron because by the process of splicing, these introns are removed. It contains only the exon. So exon appears, but introns do not appear. Option number 2 is the right answer. Absolutely correct, Karthik. Absolutely correct. Right, now moving ahead. So guys, in case if you have forgot to press the like button, do press the like button and share this video to maximum of your friends, to maximum students, so that maximum of you can avail the benefit of quality content and quality education which we are providing here on this awesome channel called Let's Crack NEET UG in English. Okay, guys. Next question. The human chromosome with the highest and the least number of genes in them are respectively. So, which one of the following human chromosome has the highest and which one has the least number of genes? So, what do you think is the right answer? Again, a simple one. We know that the chromosome with the highest number of gene is, it is a really easy one. So yes, chromosome number 1 has the highest number of has the highest number of genes, right? In chromosome number 1, there are approximately 2968 genes present. Okay, 2968 genes present. And the least number of genes is present in chromosome number Y. In chromosome Y, right? This Y chromosome, it contains only 231 genes. So it is the smallest chromosome. So the right answer is chromosome 1 and chromosome Y. So option number 3 is the right answer. Option number 3 is the right answer to this question. Okay. Question number 12. Who amongst the following scientists had no contribution in the development of the double helix model for the structure of DNA? Rosalind Franklin, Morris Wilkins, Erwin Chargaff, or Mendelssohn and Stahl? Okay. So the question is asking about the classical double helical model proposed by Watson and Crick. But I have told you how Watson and Crick lead to the discovery of the double helical model of DNA. It was not only because of their work. Their work was based on the research which was already conducted, by some of the scientists like Wilkins and Franklin. They together, gave us the X-ray crystallographic image of the DNA. Chargaff gave us the Chargaff rule, right? About Chargaff rule that the percentage of purine is equal to the percentage of pyrimidine in the structure of double-stranded DNA or double-stranded RNA. But Mendelssohn and Stahl, they proved semi-conservative method of DNA replication. They have no role in in proving the double or in describing the double helical model of the DNA. So fourth option is the right option. Absolutely correct, Karthik. Question number 13. DNA is a polymer of nucleotides which are linked to each other by 3,5 phosphodiester bond. To prevent polymerization of nucleotides, which of the following modifications would you choose? Replace purine with pyrimidines. Remove or replace third hydroxyl group in deoxyribose. Remove or replace second hydroxyl group with some other group into deoxyribose, or both B and C. So this is a good question, although a simple question. Those who already know the basic of structure of DNA will be easily able to solve this question. So we know that I have already shown you that how does the polymerization takes place. How does the polymerization takes place? Absolutely correct, Karthik. See, I told you that the phosphate which is present at the fifth carbon of one nucleotide, it attaches with the oxygen present at the third carbon of second nucleotide. Right now, if you remove this third OH group, then this ester bond will not be formed. Thus, the polymerization will be stopped. So, the right answer is option number 2. Either remove or replace this third hydroxyl group in deoxyribose. Okay. Option number 3 is absolutely wrong. The second OH group is already absent in deoxyribose. right? That's the difference between the ribose and the deoxyribose. So third option is already wrong. Option number 2 is the right answer. Question number 14. Discontinuous synthesis of DNA occurs in one strand because DNA molecule being synthesized is very long. DNA dependent RNA polymerase catalyzes polymerization only in one direction, that is 5 prime to 3 prime. It is a more efficient process, or DNA ligase joins the short stretches of DNA. So, what do you think is the right reason or the correct reason behind the discontinuous synthesis of DNA molecule? What do you think is the right reason to this question? Question number 14. Absolutely correct, Karthik. Option number 2 is the right answer. See what happened? The enzyme, that is the DNA polymerase, it has the property of catalyzing or the polymerization of DNA in only one direction, that is from 5 prime to 3 prime. Absolutely correct, Tapajit. Now, welcome to the session. Now, next question, question number 15. Which of the following steps in transcription is catalyzed by RNA polymerase? Which of the following steps is catalyzed by RNA polymerase? Initiation, elongation, or termination, or all of the above. Now, the process is transcription, that is formation of RNA from DNA. Now, in this process, which one of the following step is catalyzed by RNA polymerase? What do you think should be the right answer, guys? Karthik, Tapajit, Kunal, what do you think is the right answer to this question? So, remember, the RNA polymerase is the enzyme which is involved in the transcription process, and with the help of certain other factors like the sigma factors and the rho factors, it ensures the complete regulation in the process of transcription. Remember, the sigma factor along with RNA polymerase, it helped in initiating the process of transcription. That means yes, RNA polymerase is participating in the initiation part also. Then elongation. So elongation is the property of RNA polymerase. It is the part where RNA polymerase is predominantly functional, main part, right? Then coming towards the termination. So with the help of rho factor, this RNA polymerase is able to recognize the termination sequence also. So RNA polymerase is present here also. So the right answer becomes all of the above. The right answer becomes all of the above. No, no, Rekan, elongation is not the right answer because here the process of transcription is asked. And RNA polymerase is involved in all the three steps. Okay, the core enzyme of RNA polymerase is present, and then the sigma factor attaches to this core enzyme, and this is how the initiation started. Okay, it identifies the promoter sequence. Similarly, the rho factor, it attaches to the core enzyme of the RNA polymerase, and this is how the termination has happened. And the nascent RNA, that is the newly synthesized RNA, falls off, right? So all of the above option is correct. Now, question number 16. Control of gene expression in prokaryotes takes place at the level of DNA replication, transcription, translation, or none of the above. So, control of gene expression in prokaryotes takes place at which level in case of prokaryotes? Okay, it is a good question. We know in case of eukaryotes there are certain modification which is taken place. Okay, so in eukaryotes there are multiple steps where this gene expression can be regulated. Absolutely correct, Karthik. Right answer is transcription. Right answer is transcription. During the formation of this RNA, the gene expression can be regulated in case of prokaryotes. Whereas whether if the question would have asked about the eukaryotes, so we know that in eukaryotes the regulation can be done at multiple levels, at the transcription level, at the post transcription level, even at the translation, post translation, there are certain modifications, there are various modifications which has taken place, right? Moving ahead. Question number 17. Which of the following statement is correct about the role of regulatory proteins in transcription in prokaryotes? So, what do you think? You must have read about the operons, right? So, now this question is from one of them. So, this question is asking about the role of the regulatory protein in the transcription, and the options are they only increase, you know, they only increase expression, they only decrease expression, they interact with RNA polymerase but do not affect the expression, they can act as both activators and as repressors. You need to identify the role of regulatory proteins. Regulatory proteins are the regulators. Regulatory proteins are what? Regulatory proteins are the regulators. Okay, so student, it doesn't matter like whether you are alone or you are in a group, as long as you are sincere enough and you want to learn something. Okay, your own effort, your individual efforts, matters in your NEET UG preparation, because it is not a child's game, right? NEET UG preparation need a sincere effort, a dedicated and honest efforts, right? And a large and lot of hard work, right? A lot of hard work. There is no shortcut to success, right? So only those students who are sincere enough and who feels like yes, this is an important part of their preparation will definitely be finding time to participate in the practice sessions, right? Because there is no other distractions available here. For this entire 1 hour, we are solving questions, we are discussing the logic behind it, and then we are leaving, right? So tell me the answer for question number 17. See, the regulator proteins which are available, they perform both the functions. They can either activate or they can either repress the transcription in prokaryotes. And you have seen the two operons, right? The trip operon and the lac operon, right? You know the that in both the cases, the both were the examples. One was the activator and one was the repressor, right? So fourth option is the right answer. Moving ahead, question number 18. Which was the last human chromosome to be completely sequenced? And I think you already know the answer. Okay, I have already told you that the chromosome 1 is the chromosome with the maximum number of genes. I told you that 2968 approximately 2968 genes are present on this chromosomes, and this particular chromosome is the chromosome which was last to be sequenced and was sequenced at around May 2006, right? So the right answer is option number 1. Question number 19. Which of the following are the functions of RNA? It is a carrier of genetic information from DNA to ribosome, synthesizing polypeptides. It carries amino acids to ribosomes. It is a constituent component of ribosomes, or all of the above. What do you think is the right answer, which explains the function of RNA in a correct way? So, which of the following are the functions of RNA? So yes, all the three statements are correct, because the information from the DNA is transferred to RNA, and and this information is involved in the synthesis of protein. So first option is absolutely correct. Yes, absolutely correct, Karthik Kunal. Option number 4 is the right answer. It carries amino acids to ribosomes. Yes, tRNA does the function. It is a constituent component of the ribosomes. rRNA does the function, right? And here mRNA is there, right, in this question, in this part. So option number 4 is the this is option number 4, right? Option number 4 is the right statement. Absolutely correct. Now question number 20. While analyzing the DNA of an organism, a total number of 5386 nucleotides were found out, of which the proportion of different bases were adenine 29%, guanine 17%, cytosine 32%, thymine 17%. Now considering the Chargaff's rule, it can be concluded that it is a double stranded circular DNA. It is a single stranded DNA. It is a double stranded linear DNA, or no conclusion can be drawn. So, what do you think is the right answer to this question? There are various percentage of the composition of the nitrogen bases are given. But as far as I remember, and as far as you have studied, according to Chargaff rule, so first of all, this Chargaff rule is applicable only to the double stranded, DNA or RNA molecule. Okay, let us say if it is a double stranded DNA. So according to the Chargaff rule, the percentage of purine is always equal to the percentage of pyrimidine. That means adenine will always be present in equal percentage as that of the thymine, whereas guanine will be present in equal percentage as that of the cytosine. But here we see that adenine and thymine are not present in equal percentage. Adenine is present only 29%, whereas thymine is present only 17%. So that means they are not equal. Similarly, guanine is present only 17% and cytosine 32%. They are again not equal. That means it is a single stranded DNA. It is a single stranded DNA. Very good, Karthik. Now, question number 21. Question number 21. If in some viruses, DNA is synthesized by using RNA as template, such a DNA is called. Okay, so DNA where, when RNA acts as a template in the synthesis of DNA. Okay, that means when the information moves in the backward direction. We know this process is known as what? This process is known as reverse transcription, right? So the DNA which is formed over here, when RNA is acting as a template, but the process of reverse transcription, this DNA is known as aDNA, bDNA, cDNA, or rDNA. So the right answer to this question is option number 3, that is cDNA. Okay, it is the complementary DNA. cDNA stands for complementary DNA. Complementary DNA. Okay. Next question. Question number 22. Michelson and Stahel experiment is continued for four generations. So four generations in bacteria. Now the ratio of N15, N15, N14, N14 and N14, N14 again containing DNA in the fourth generation would be. Okay. So that means a DNA molecule, I'll be drawing it over here. See what happens. A DNA molecule is given to you. So this DNA contains one strand of N15 and another strand of N14. Now, this DNA molecule is allowed to replicate for fourth generation. Example, I won't be drawing it for the fourth generation. I'll be giving you time to calculate it. Now, see, in the first generation, so we know that DNA replication is semi-conservative. That means both these strands will separate and behave as the template. So one is N15, another is N14, right? And medium is what? Medium is the normal medium, that is N14 medium. So now the next DNA strand will be synthesized, will be of N14 nature. Similarly, the next here will be of also N14 nature. Okay. Now again for the second generation, when the second time when this DNA will be dividing or replicating, again the four strand of the DNA will be behaving as the template. So separate all the four strand and label them. One is N14, another is N15. Rest two are what? N14, N14. Right. Now the next new strands which will be synthesized, all will be of N14 types because the medium is what? N14. So rest all will be N14 types. So you can see that as you are progressing towards, the next generations, as you're progressing towards the next generations, you are seeing that, N15, N15 condition is not found. N15, N15 is not found, right? N15, N15 is not found because, every time medium N14 is present. So N15 and 15 condition is not present, not possible. So it will be 0. It will be how much? It will be 0. Right. Now you can see in this generation you have how many molecules which contain how many strand of the DNA which contains N15? Only one strand. Only one strand. In the third generation also, this N15 strand will be there. It will be, forming N14, N15 DNA, and rest all the N14 type will be forming N14 DNA. Right. Now in the fourth generation again, you are left with only one strand of N15. Only one strand of N15, but rest all the DNAs are of N14 types. So the right answer comes to be option number 4. Absolutely correct, Rekan. Right answer comes to be option number 4. And this is the method how you solve this question. Right answer is option number 4. Okay. There will be 7 DNA molecules with N15 and N14 combination, and there will be 14 DNA molecules with N14 N14 combination. And when you simplify it, the ratio becomes 0 is to 1 is to, right? There will be not 7, there will be 1, and it will be 7 molecules, N14 and N14 one, right? So this is how you find out the molecules. Now moving ahead, if the sequence of the nitrogen bases of the coding strand of DNA in a transcription unit is 5 prime say 3 prime is given, ATG, the sequence of bases in its RNA transcript is. So 5 prime to 3 prime sequence is given. Now you have to identify the sequence in its RNA transcript. Now let's see who gets it right. This is a really good question. Now I want you to attempt it right? So what is the right answer to this question? Very good, Karthik. Very good. Really appreciable. So, this 5 prime to 3 prime strand is the coding strand, remember? So, the information which has been passed to the RNA transcript with the help of the template strand will be exactly the copy of the coding strand, and that is why this strand is known as the coding strand. Okay, that is the only reason why this strand is known as the coding strand. But in place of thymine, uracil will be present. So, that's the only difference. So, see, A, then uracil, then guanine, then again uracil in place of thymine. So right option is option number 1. Very good, Karthik and Rikan. Question number 24. The RNA polymerase holoenzyme transcribes. Okay. The RNA polymerase holoenzyme transcribes the promoter, structural and the terminator region. The promoter and the terminator region, the structural and the terminator region, or the structural gene only. So guys, welcome to the session who are joining our session right now, and try to participate in the questions, try to solve them, try to write, give the answers. So the RNA polymerase holoenzyme transcribes which one of the following part? Does the promoter, structural and terminator all the 3 part, or structural and terminator, or only the structural region? Remember, this RNA polymerase, with the help of the sigma factor, it identifies the promoter region. And when it binds to the promoter region, it just initiates the process of transcription. But it do not transcribe the information which was present in the promoter region. The promoter region binding to the promoter gives it a hint that yes, the structural gene is present near about it. Then it moves towards the structural gene. There the process of transcription takes place, right? Then what happens? It encounters the terminator region with the help of the rho factor, and as soon as it encounters the terminator region, the newly formed RNA falls off, right? That means it is transcribing only the structural gene, not the promoter and the terminator. Promoter is to help in initiating the process, and terminator is to help to stop the process, right? So the right answer is option number 4. This was a good question. Yes, Karthik. So, Rekan, Karthik, absolutely correct. Now, question number 25. If the base sequence of a codon in mRNA is 5' to 3' AUG, the sequence of tRNA pairing with it must be. The sequence of tRNA pairing with it must be. We know that, the codon is present in mRNA and the nodok or the anticodon is present in the tRNA, right? That means its complementary sequence becomes what? UAC. UAC. Its complementary sequence becomes UAC, and ends becomes also complementary 3' to 5', right? But now the options are given from 5' to 3'. So if you read this information from 5' to 3', so information becomes, see, this is the mistake which we generally do. You have to read the information from 5' to 3', because this is how the options are given here. So reading the information from 5' to 3', it becomes CAU. It becomes CAU. So the right answer becomes option number 2. So this is a good question, right? This is a good question. See, UAC is the 3' to 5' direction. All of you are doing the same mistakes. I hope now you got it, right? So the next question, question number 26. The amino acid attaches to the tRNA at its 5' end, 3' end, anticodon site or DHU loop. So at which particular site is the amino acid attached? At which particular site is the amino acid attached to the tRNA? We know there are approximately 4 loops present in the structure of tRNA. First, or the DHU loop. DHU loop, amino acyl synthetase recognition, right? Then anticodon, it helps in the recognition of the mRNA, reading the codons, right? Now coming towards the 5' end and the 3' end, excuse me, polyguanine sequence are present at the 5' end and the amino acids are attached at the 3' end, where this sequence is found, right? So it is attached at the 3' end. Okay, so second option is the right option. I think most of you got confused. So this is the right answer. Question number 27. To initiate translation, the mRNA first binds to the smaller ribosomal subunit, the larger ribosomal subunit, the whole ribosome, or no such specificity exists. So in order to initiate the translation process, the mRNA molecule will bind to both the subunits at the same time, or will be, binding to different subunit. And if it is binding to different subunit, then what is that subunit where mRNA is binding? So I have told this multiple times. Very good, Karthik. Right option is option number 1. The mRNA binds to smaller ribosomes. And when multiple ribosomes, when it comes attached to the mRNA, right? This structure is known as what? This structure is known as polysome or polyribosome or ergosome. Remember? So first it binds with the smaller subunit. Question number 28. In E. coli, the lac operon gets switched on when lactose is present and it binds to the repressor. Repressor binds to operator. RNA polymerase binds to the operator. Lactose is present and it binds to RNA polymerase. So a question from lac operon. Okay. So example of inducer operon. So you have to identify the correct answer with respect to the E. coli operon, E. coli lac operon when lactose is present. So what is happening? Generally, the repressor protein is present and it is binded to the operator region, and that is why operator is not able to proceed with the process of transcription. But when lactose is present, it binds with the repressor. Thus, the repressor is not will not go and bind to the operator. The operator becomes free, and this is how the operator become functional. And this is how, in the presence of lactose, the induction of the lac operon takes place, and that is why lactose is known as the inducer. So, option number one is absolutely correct. Very good, Rekan and Karthik. Absolutely correct. Now, question number 29. In eukaryotes, RNA polymerase 3 catalyzes the synthesis of. Now you have to identify the synthesis which is catalyzed by RNA polymerase 3. All are RNA and tRNA, mRNA, hnRNA or snRNA, 5s rRNA, tRNA or snRNA, 28s, 18s or 5s rRNA. So what is the right answer to this question? RNA pol 1, RNA pol 2 and RNA pol 3. We know there are three RNA polymerases, right? So do you remember what are synthesized with this RNA polymerase? 1, rRNA. RNA polymerase 2, mRNA. Also hnRNA, heterogeneous nuclear RNA, the pre-mRNA molecule. RNA polymerase 3, tRNA. And also snRNA. And also the 5s rRNA, which is not synthesized by RNA pol 1. So the right answer is option number 3. Right answer is option number 3, 5s rRNA, tRNA and snRNA. See, this is the sequence: RNA pol 1, 2, 3 and RMT. RMT, right? So like PMT, it is RMT. So RMT is the sequence, and apart from this you know that hnRNA is the preformed mRNA, so RNA pol 2 is catalyzing it. Okay. Now we know that all the type of rRNA are synthesized inside the nucleolus except the 5s rRNA. Okay, this 5s rRNA is synthesized outside the nucleus but inside the outside the nucleolus but inside the nucleus, and by the enzyme RNA polymerase 3. Okay, and snRNA, which is involved in the process of splicing. So right answer is option number 3. Question number 30. First evidence in the favor of DNA as genetic material comes from the experiment of Griffith effect, conjugation, or Taylor's experiment, or Kent's effect. So, there were multiple experiments which were conducted by a group of scientists in that era to prove that yes, DNA is the genetic material, and at the same time to disprove that protein is not a genetic material, because many group of scientists at that time used to believe that since protein is a diverse molecule, it is made up of 20 different types of amino acid, definitely it is a it must be acting as a genetic material. But the series or the sequence of the experiments which started, it started from Griffith. Okay, first of all, Griffith transformation principle came, which gave us the hint that the bacterial cell is transforming. That means something is transferred, right? Then multiple experiments were done, like for example the biochemical experiment conducted by the the Avery, McLeod and McCarty. Then Hershey and Chase experiment. So the first was the Griffith. So right answer is option number 1. Okay, very good. Now question number 31. So some questions from previous year examination. So some question from previous year examination. DNA fragments are, it has been asked in NEET 2017, negatively charged, positively charged, neutral, or depends on the size. So what do you think is the charge on the DNA fragment? So this type of question we have already solved in today's practice session. So I hope each and every one of you gets it right. So the right answer is negatively charged. Yes, because of the presence of phosphate ion. Absolutely correct. Now, if there are 999 bases in an RNA that codes for a protein with 33 amino acids, and at the base position 901 is deleted, and and the base at position 901 is deleted, such that the length of the RNA becomes 998 bases, then how many codons will be altered? Then how many codons will be altered? So, this question has been asked in NEET 2017. What should be the right answer to this question? See, we know that the nitrogen bases are present in the in the RNA molecule. For example, if this is the sequence, okay, if this is the sequence. Now what happens? Now what happened at 901? At 901 position, a particular position at particular base is deleted. For example, if this is deleted, so initially what happened? The triplet was like this. But now if we delete this one, So now what happened? Now if we delete this one, so the codon will now disturb. The codon will now, the genetic code will be reread in, the different triplets will be formed. So that means all the different, the 33 codons will be altered. The different 33 codons will be altered because at the 901 position, at the base position at 901. So before that position, all the amino acids, all the codons were right. Okay, all the codons were right, but after that position, that means around 98 bases, these 98 bases will be reread, right? And we know that 3 nitrogen base, it constitutes one codon. So approximately how many codons comprises this 98 bases? Approximately 33 codons. So the right answer is option number 4. Okay, right answer is option number 4. Question number 33. Which of the following rRNA acts as the structural RNA as well as ribozymes in the bacteria? NEET 2016. 23 sRNAs, 13 sRNA, 33 sRNA, 73 sRNA. First of all, this question, if you would be, attempting this NEET 2016, this question was really easy because you know the different types of RNA, okay, in the bacterial ribosomes. Okay, so first of all, we have never seen this 13s rRNA, we have never seen this 33s, we have never seen this 73s. So we already know the answer. The right answer is 23s. This question would have been difficult if all the right options were given. For example, 23s, 5s, okay, then 16s, and let's say 18s also was given. In this case, what happens, we know that out of these four, these three are present in bacteria. And now, in case if you don't know the answer, then you might get confused. But we know that 23s RNA has the ribozymal activity. So right answer is option number 1. Question number 34. The equivalent of a structural gene is NEET 2016 question, right? So this structural gene is also known as what? Operon, muton, cistron, or none of the above. Okay, or none of the above. So we have seen the operon model in the gene regulation part in the prokaryotes. Muton is the minimum unit in which mutation can take place, and cistron is the minimum sequence of the information which contains information for a polypeptide synthesis. That means a structural gene is equivalent to a cistron. Right option is option number 3. The structural gene is equivalent to a cistron, right? Moving ahead, what happened, guys? You got confused or what? This is a easy question, right? Question number 35. Now, Taylor conducted the experiments to prove semi-conservative mode of chromosome replication on Vixia faba, Vinca rosea, E. coli, or Drosophila. So now you have to tell the experimental object which was used by Taylor in proving the semi-conservative method of chromosome replication. So again, a direct question from the theory part. You must get it right because see, it is an easy question. If this question comes in NEET 2021 or 2022, you must be able to solve it. What is the right answer to this question? Vixia faba. Vixia faba was the experimental subject which was used by Taylor. Right? Very good, Rikan. Now, question number 36. Again, NEET 2017. So, which of the following RNA is the most abundant in animal cell? tRNA, rRNA, mRNA, or none of the above? Most abundant RNA you have to determine. I think when I was discussing biomolecules in my yesterday's class only, I told you this order of abundance of RNA. Okay, the order of abundance of RNA. So, what is the right order? What is the right order, students? I'll be writing 3 RNAs over here so that I'll be giving you some time to think. Now, I have to decide like what I have to place. So, the most abundant RNA, approximately 80% of total cell RNA. Then approximately 10 to 15% of total cell RNA. And here it is approximately 1 to 5% of total cell RNA. Okay. And the most abundant RNA is the rRNA, followed by the tRNA, followed by the mRNA. So the most abundant option becomes option number 2, that is the rRNA. So this time you was wrong. Second option is the right option. And this is the order of abundance of the RNA in the cell. Okay. Next question. Question number 37, again of NEET 2017. The association of histone H1 with a nucleosome indicates DNA is condensed into chromatin fibers, transcription is in progress, DNA replication is in progress, or DNA double helix is exposed. Now, what does the association of H1 histone with a nucleosome indicates? So, till the time you are thinking, I am drawing the structure of the nucleosome, right? So remember the nucleosome. Nucleosome is an octamer. Nucleosome is an octamer. And DNA thread is coiled around it. And with the help of the linker histone, which is the H1 histone, it is connected. Right now, the question is saying that the association of histone H1 with a nucleosome indicates. Now, this H1 is associated with this nucleosome, okay, which means it contains 8 histone protein along with the DNA fragment. Right. So, there are four options. DNA double helix is exposed. How is this association helping us in, how is this association helping us in this part? No, this is not correct. DNA replication is in progress. No. Transcription is in progress. No. DNA is condensed into the chromatin fiber is the right option because it is associated with histone, H1 histone. Okay. So DNA is condensed in the form of the chromatin fiber. Remember, chromatin fiber is the example of nucleoprotein, one of the category of the conjugated protein where the prosthetic group was the nucleic acid, right? So first option is the right option. Question number 38. The final proof for DNA as a genetic material comes from the experiment of McLeod and Avery, Griffith, Harvey McBride, or Hershey and Chase. So the final proof for DNA as a genetic material came from the experiment of which one of the following scientists? What do you think? What do you think? First of all, Griffith transformation principle was the first. Now, McLeod and Avery and McCarthy also, they did the biochemical analysis. You remember? Okay, they already proved that yes, DNA is a genetic material with the help of the biochemical analysis. But their work was not that recognized. And they were deserving a Nobel Prize for this discovery, but they never received this Nobel Prize. This is one of the biggest omission by the Nobel Committee. Okay, by the Nobel Committee. Okay, so then the observation of McLeod and Avery were finally proven by Hershey and Chase when they used bacteriophage to infect the E. coli bacteria. And what did they do? They radio labeled the phosphorus and the sulfur. Remember, the radio labeled phosphorus and radio labeled sulfur was used. And then the infection of the bacteriophage was checked. So, right answer is option number 4, Hershey and Chase. The transduction experiment, right? This was the transduction experiment. Now, next question, question number 39. Spliceosomes are not found in the cells of bacteria, animals, plants, or fungi. So, spliceosomes are the complex which are involved in the process of splicing. It includes certain proteins along with the snRNA, that is a small nuclear RNA, which is synthesized by RNA polymerase 3. So it is not found in the cells of. So fungi, plantae, animalia, all these three different examples comes from the eukaryota. Okay, they are all eukaryotes. Only bacteria is the member here which is prokaryotic in nature, and we know in bacterial cell splicing does not takes place. Bacterial RNA contains only the functional parts. So right option is option number 1. Right option is option number 1. Okay, what happened? Did you guys find it difficult or what? I didn't found any answer. So now next question. Last question for today's session. The commonly used vectors for human genome sequencing are TA cloning vector, expression vector, tDNA, or BAC and YAC. So BAC and YAC. So who knows the full form of this BAC and YAC? This is tumor DNA, right? So what are the commonly used vectors for human genome sequencing, guys? This was asked in NEET 2014. Well, our target is to score 360 out of 360 in biology. Is it possible? Do you guys think that yes, you'll be able to score this score 360 out of 360? I want to know your answer, because each and every individual or each and every aspirant who is planning to who is planning or preparing for NEET UG examination and is sincere enough and working really hard can definitely score this much. They can easily score more than 340 marks. And I say why 340? You have to score 360 out of 360. And yes, it is possible, because my dear students, you can see the paper and you can compare the papers of various of past 5 years or past 6 years. You'll realize that each and every question which has been asked in the NEET examination comes directly from the NCRT, right? So it is either directly from the NCRT or it is related, somewhat related to the NCRT. So if you have conceptual clarity, if you have practice questions from NCRT exemplar also, and okay, and previous year questions and also NCRT based questions, and definitely it is possible to get this score, because we do not score well because we end up doing silly mistakes. What happens in biology, mostly the question level is easy, so we get overexcited and we end up doing the silly mistakes, and this is why we are not able to score good. But we have to improve on this matter, right? So yes, option number 4 is the right answer. BAC stands for bacterial artificial chromosome. Bacterial artificial chromosome, right? And YAC stands for yeast artificial chromosome. And yes, they are commonly used vectors for human genome sequencing. They have quite a good capacity to hold gene of larger size. Option number 4 is the right answer. Absolutely correct, Rekan. Absolutely correct. So guys, that's it for today's practice session. So I hope you guys enjoyed a lot and you learned something new. Okay, so before taking your leave, let me again tell you my timing. I'm live daily at 5 p.m. on Let's Crack NEET UG in English, your one and only favorite channel for your NEET UG preparation, right? Where we are regularly providing you live lectures and important updates related to NEET UG examination. If you follow them genuinely, you will be definitely scoring really good marks, right? Then I'm live at 9 p.m. for practice session. I'm live at 9 p.m. for practice session, okay? And these practice session are for your problem, for enhancing your problem solving ability, okay? I'm live again at 11 a.m. on Unacademy learning app. You can follow me on Unacademy Learning app to get all the updates regarding my class timings. Okay, guys? And in case if you have still not subscribed for Unacademy Plus or Unacademy iconic platform, so do subscribe and start your preparation soon, ASAP, right? And do not forget to use my referral code AG001 to get an instant 10% discount. Okay, guys? And still, if you have not, press the like button and share this video. So do press the like button and share this video to the maximum. Thank you. Have